I'd like to introduce our next speaker, Andreas Stahl, here at UC Berkeley. And Andreas, I'm going to give you the microphone. And again, keep these away from one another. Fantastic. Again, right here, keep them separated. Thank you.
Very good. Got to keep them separated. Well, thank you all for staying here. I know the lunch is waiting. I'll try and be concise. What I want to talk to you today is going to bring together some of the topics that we actually heard about. I'm going to try and synthesize some of them, including the use of stem cells in aging research, stress tests, the importance of biological variables as well. So my lab here is at UC Berkeley. I'm trained in metabolic biology, but we've been segueing both into the stem cell as well as the aging field. And what has really been interesting us a lot is driven by this chart here, which of course many of you have seen already, which is the failure in the drug pipeline that we are seeing, where we're starting out with thousands, tens of thousands of ideas and ultimately end up with 0 to 1 drug. This is driven by many factors, but in part it is driven, as we're realizing now, by the use of preclinical model systems that do not reflect human biology correctly. That realization has led to initiatives both at the level of NIH, FDA, and EPA that are now trying to emphasize the use of so-called new approach methodologies that combine both the use of advanced in silico approaches as well as new in vitro approaches, and it's the latter that I want to talk to you more about today. So how can we look at human diseases? How can we look at human health? How can we look at human aging using a complex human-based preclinical platform? Now, the answer to this, I think, in my mind, lies in building complex systems that recreate inter-organ communication, which is really at the heart of what disease pathologies are often about, using new tissue culture approaches. You're all familiar with the classical approaches where you have cells sitting in a dish here, two-dimensional, sitting on a really stiff substrate. None of this is in any way physiological. It's sitting in sugar water as well. This has been tried to make more complex by culturing 2 different cell types in the same well, but really hasn't added much complexity. Now, there's a new field called organoid biology, where people are using often stem cells, but also primary cells to cluster them, to recreate organoids. This has been mostly successful in the case of liver organoids as well as intestinal organoids. But it has become very difficult to look at inter-organ communication using these organoid systems, because you can not really culture many of them together. So how can you do this? The answer to that is shown here, and this is known as the organ on a chip or microphysiological platform that allows you to culture cells in a 3-dimensional environment with flow characteristics that you would get inside of the human body. And indeed, you cannot just have one tissue in these, but you can multiplex different tissues and bring together a multi-organ system to then investigate human biology. You can use this microphysiological system, as I said, for many purposes, and they have many advantages. These are actually some that we are making right here in this building, where there's a core facility produce these microfluidics. And their advantages are that you can easily scale the cells. You have full control over the diffusion and perfusion conditions. There's much that I won't have time to get into, but if you want to know more about the details, it's great. They are highly scalable, miniaturizable. They consume minimum amounts of fluids that you pump through these systems. And we decided to use these in combination with iPS-based systems. We heard about stem cells, we heard about direct differentiation of cells. We are using here iPS-derived cells for several reasons. One of them includes that they are genetically trackable. There's a plethora of cell types that we have now high confidence that we can differentiate these cells into. And what we've been focusing in my lab particularly is to take these stem cells and differentiate them into tissues that drive metabolism in the human body. And the core tissues that you have to think about when you talk about metabolism is the fat-liver axis. That's the communication in your body that regulates the switch between fasting and fat states, where you either store energy or you consume energy. These are really the two metabolic hubs. But this is an aging conference. There's also a reason to look at fat in the context of aging. We already heard from Dr. West the idea of brown fat decline with aging. There are also significant changes in white fat with aging. In fact, it's one of the early tissues that changes with aging, and it has a myriad of knock-on effects on other tissues, including the liver, and ultimately gives rise to many of the complications that lead to the leading causes of death, as well as chronic diseases listed here. These are all really driven by adipose tissue liver dysfunction. How can we look at that with the microfluidic devices? So we have now optimized differentiation protocols for the iPS cells to create adipose tissue. These here are the white adipocytes. They are stained for their lipid droplets, and they form these nice three-dimensional clusters. You can see here, this is about the real size. They are very small wells. And we can integrate this with iPS-derived hepatocytes. Importantly, these are derived from the same iPS line. So everything I'm going to show you here is isogenic, meaning we just heard about the importance of genetics. These all have exactly the same genetic background, and we're fully able to manipulate these genetic backgrounds as well experimentally, but I won't have time to show you that. So these two systems can be interconnected. You can play with the scaling, which then can give rise to saying that you would have an obese state or a lean state, right? If you look at the number of adipocytes per hepatocytes, and you can further make this system more physiologically relevant through the introduction of immune cells. In this case, what I'm showing you here is that we are flowing in macrophages that then migrate into this adipocyte cluster. These are big cells, so you can't really see through it. But these macrophages, too, are differentiated from the same iPS cells. So this is, again, an isogenic system. There is no immune rejection because of different HLA phenotypes here between the different cell types. And they migrate into these fat cells. And you can do this with pro- and anti-inflammatory macrophages. And you can study, for example, fatty liver disease in a system like this and try and explore the effects of drugs on the development of fatty liver disease. As you see, in many people that are obese, 1 in 3 Americans suffers from fatty liver disease, and you can try and look at insulin sensitivity. We heard very early in the first day about the importance of diabetes and how incredibly common it is in many countries, including Saudi Arabia, which I believe is still in the top 10 with some 20% or so, right? Go Saudi Arabia. Yes, so how can you then use these systems to look at biological variability, which is really what we also heard about earlier on when we talked about menopause. This is that although we have a defined genome here that we can look at, right, the genetics, there are 2 other forms of variabilities that are almost always excluded when you hear stem cell researchers talk. One is biological sex, and ask me about how we are working on that. And the other one I want to talk today about, because this is an aging conference, how can you look at the biological variable of age? Now, we heard in our talk before that from the speaker from the Buck Institute, which was an awesome talk, that the problem with induced pluripotent stem cells for aging research, and so goes the general wisdom, right, is that when you make these cells, that they represent a neonatal state, right? That you end up with an immature hepatocyte, that you end up with a fat cell that looks like the fat from a baby, right? So how can you use this system and age it? And there's been different approaches that people have taken. This includes irradiation and Ross treatments and some cocktails that may induce senescence, but none of these really recreate any kind of physiological aging stage. And to be frank, without someone sitting right now in the audience, we would have never thought about using this system for aging, but Irina Conboy convinced me that this can be used to look at aging, Andreas, using an approach that she had pioneered, which is based on this really cool finding in heterobiotic mice where, and I'm going the wrong way, where you can find that if you stitch together, and Michael Comboy showed this too on the first day, where you can stitch together two mice, an old and a young one, and the young one gets a lot older and the old one gets a little bit younger. So that means that somehow in the serum there are factors that we still don't fully understand that drive aging phenotypes. And in the mice, they can do this very fast. This also has been done with primary cells by many different labs and reproduced over and over and holds to be true. So our idea then was, can we actually use the system of organ-on-a-chip-based human tissues, interacting tissues like fat and liver, to recreate a state that would equal an aged human state and a young human state from exactly the same cells. That's something that's really hard to do with a direct differentiation or transdifferentiation approaches, because you don't have fibroblasts for a lot of people that were young and then give 80 years again the same fibroblasts, right? So here we have the same cells and we want to recreate both the young and the old stage. And the way that we're doing this is shown here in a schematic where we are starting out with one IPS line, and we've done this now with many, but we always keep it consistent between the different cell types that we create, and we make either adipocytes out of these or hepatocytes, and we have specialized devices for both of these different cell types, and then we can interconnect these devices or interrogate them separately. And the way that we are inducing the aging phenotypes is by adding 5% human serum from either donors that are in the old category, which was 62 and older years, or in the young category, which is around 21 to 30 years of age. We flow this serum through our devices for 4 days. We're not waiting for 2 years like with a mouse. We're not even waiting for months as you would with a worm. 4 days. Then we do a lot of phenotypical assays, not just transcriptomics. We can do proteomics. We can look at inflammation markers. We can do functional assays, such as insulin sensitivity assays. And this is what you get. And I'm sorry, this is a lot of data. I will try and make this as simple as possible. So when we look at senescence, this here is the famous beta-gal staining. These little round dots, these are the adipocytes inside of the microfluidic device. That was the first result my postdoc brought me, where he held up the chip, the upper row was clear, the lower row was blue, and he said, guess which one had the old serum? Well, it was this one here, right? And what you can see is that there is a massive induction of senescence, and in fact, both of the genes you heard about, P16 and P21, are induced, so there's robust induction of senescence, and concomitant with that, there's also cell cycle arrest induction, which is again interesting. Just like with the neurons, what you heard before, these cells are already cell cycle arrested, but still you get massive induction of more genes that are related to cell cycle. There's also huge induction of pro-inflammatory factors that are also known as SASPs. And then I think another completely unexpected result is that within these 4 days of exposure to old serum, but not with the young serum, you get induction of highly significant DNA damage in these adipocytes on a chip. You also get a change of the markers for what kind of adipocytes you're making. I won't take you through what all these genes are, but you're all familiar with the difference between the beer belly fat, right? This is your visceral fat, and your subcutaneous adipose tissue. There's a huge difference in terms of the biology. The intra-abdominal fat is highly detrimental for your physiological health and for your metabolism, while the subcutaneous adipose tissue is much better tolerated. When we originally make these adipocytes from iPS cells, we make fat that looks very, very subcutaneous-like. When you expose the subcutaneous fat to the serum from old donors within 4 days, you see the induction of markers for visceral fat, which means that we're giving these chips a beer belly, basically. And coming back to functional assays, we heard about diabetes. Insulin resistance is a hallmark of type 2 diabetes. We also see the induction of insulin resistance, which is something that declines with age as well. Insulin sensitivity keeps going down as we age. Another hallmark here specific for adipose tissue that also happens. Similarly, and I won't take you through all of the details, this was just recently published in a paper by Irina's lab and mine in Nature Biomedical Engineering. These are now the hepatocytes, the same assays that I showed you. Again, robust induction of senescent cells. You see that there is cell cycle arrest. You see that there is senescence-associated protein expression. And important for the liver, I started out by talking about fatty liver disease. This too is not just associated with obesity, but it's highly associated with aging as well. And we see, indeed, after 4 days of treatment with serum from old donors, that there is an induction of markers that you would find in fatty liver disease. So many hallmarks of aging here, more than I can show you due to time, but are these really replicating what you would see in human adipose tissue in terms of the transcriptomic profiles? The answer is yes. What we did was a really unbiased approach. And I got to speed up here a bit. We looked at the top 10 Go categories for old versus young fat in a GTEx database, which is human adipose tissue from different age groups, and compared that to our chips from the old serum, and the top 10 categories are exactly the same. There are other ways of measuring this. I won't have time to talk to you, but the take-home message is always the same, that we recreate the hallmarks of human aging for the tissues that we have. More than that, a very talented graduate student in my lab, Yu-Chen He, also developed a machine learning algorithm that can take these GTEx data and parse them more by age to develop an age prediction platform, prediction of biological age here, for adipose tissue that was trained with the GTEx data, achieved very high accuracy. And again, we're actually parsing male and female here. Ask me about that later. We can do sexual dimorphisms as well. And then we can ask with our samples, well, not just do we have hallmarks of aging, but how old does this chip look to you, machine learning algorithm? And the answers are, if we have the serum from young donors, it's somewhere between 30 to 40, and it's 50 to 60 and older for our old serum exposed ones. And we've verified this more, and we've actually honed this algorithm more, and we get even better result predictions by now. We can use this also to look at markers of aging. So this here is a prediction of biomarkers for aging and pathway analysis coming from the chips. So we can use this to identify new markers for aging, but also new signal transduction pathways that seem to underlie the biology of aging that could then be targeted. Can we use this to look at rejuvenation approaches? Absolutely. So we've done a lot of this. I won't have, again, time to show you all. Here's some of the best data that I can show you, which is where we basically mimic what was done with the mice, where you first establish an aged phenotype, 4 days serum from old people, and then switch it to 4 days serum from young people. And indeed, you can reverse some, but not all, of the phenotypes of aging here. We have done this with anti-senolytics. We've done this with rejuvenating compounds. We get quantifiable results back. How much rejuvenation do you get? We can do this in 8 days, okay? 8 days. And we get results that are highly aligned with clinical results. For example, the mTOR inhibitor rapamycin, there we go, was taken by some biohackers by themselves, turned out to not extend lifespan at all. In our system, does nothing to improve aging parameters. We also can modulate gene expression in these devices as we go through gene knockdown. That can be done inside of the microfluidics. We can say, oh, our hypothesis is gene X drives the aging when we expose the cells to old serum. We can knock down gene X before we expose the cells, after we expose the cells, get readouts within days. Where are we taking this? As promised, I'm wrapping up here. So we have more systems between the fat, besides the fat and liver in the works that are already running in the lab. This includes our systems for obesity, diabetes, fatty liver disease, to which we now added vasculature. We have a knee joint chip now that has cartilage, bone, and adipocytes to look at osteoarthritis and the impact of aging and anti-aging strategies. And we have a bone marrow chip, really exciting, that's running now, where you can look at the shifts in the immune system that we heard earlier about. For example, what are the differentiation outcomes of hematopoietic stem cells? All of this iPS-driven, all of this able to genetically manipulate the cells. So our applications in the end are listed here. So we are very interested in testing further anti-Gerontologic treatments for screening and assessment of rejuvenation effects. You can use this to do biology, find new and test new mechanisms for aging and off aging. You can interrogate the effects of the human genome here by having iPS cells from centenarians, from people with premature aging. You can recreate your own genome by using CRISPR and changing the genes that you think are driving the phenotypes to test the hypothesis. And then you can actually also look at the changes in the effects of drugs that we use in the clinic right now, how they change when they are given to old people, because clinical trials are done with young people. So if you have someone who is 60-year-old or 70-year-old and gets a treatment prescribed to treat diabetes, that the ideas of how to treat their patients were developed from someone who was 18 or 21 years old. Okay? So that may not make sense. So we can test how well these drugs work. I want to leave you with showing you my lab here, and yes, that is a brewery in the background. The postdoc who did a lot of this work here is Lin Chi, who's right here, who now is going to start a professor position in China. And as I mentioned, without Irina Conboy, we wouldn't even have launched into this field of aging. So I'm really thankful for her to be here today. Thank you. Happy to take your questions.
Oh, yeah. Thank you very much, Andreas. Thank you so much. All right, there you go. Thank you so much. Okay, any questions for Andreas? Yes. All right, here we go.
Thank you. This is a fascinating field. I have a twofold question. First of all, you are using various techniques to essentially induce senescence in these organoids. What would prevent you from, say, taking cloned cells from elderly donors and generating the organoids that way, because then you know you've captured all of the aspects of senescence? And the second part of my question is, could there be some types of damage involved in senescence that happen in humans over time, but that your techniques aren't able to replicate quite yet? So if you reverse something in an organoid, you might not yet be able to reverse those types of damage.
Great question.
Yeah, thank you. So the first question is, why not use cells from actual old and young people? And the answer to that is because of diversity between people, right? In this case, we actually have an old and a young phenotype that is derived from exactly the same genotype. So we have a direct ability to compare this. If you take cells from an old person, you would have to take thousands of samples before you reach statistical significance. The other answer is that when you look at internal organs such as the liver, the accessibility of these cells becomes very limiting. Few people object to having a little bit of fat removed, but once I try and go down into your liver, there may be some objections to that. And of course, there's some tissues like the brain or the heart that are almost impossible to biopsy. So the supply of these primary cells with the right age type is really limiting. So those two confounding factors are the reason that we don't do it.
Great, thank you, Andreas. Great answer. Any more questions for Andreas? Going once, going twice? Lunchtime. Oh, sorry. By the way, I want to say thank you to Irene and to Michael for providing lunch today. It's very, very healthy, vegetarian. Irene, did you want to say something? Yeah.
I want to mention. For Andreas, it's like wonderful talk. First of all, he's looking to start biotech on this technology. And secondly, the age of the donor of the blood serum controls in 4 days the age of the tissue. So please think about it with respect to what is aging. We have brand new cells just made from stem cells. And the age and become in their biological age very, very similar in 4 days to the age of the donor of the blood serum, where in vivo it will take 70 years. So that opens a whole million of new questions and possibilities. I just want to
Thank you.
Yeah.
No, thank you very much, Irene. Thank you so much. Okay, I think we're done for. Oh well, one more question. Okay, we'll take one more, and then we can go for lunch. Here we go. Greg, go ahead.
So, and if you can induce just by your old blood phenotype an aged state, what does this say about trying to put in youthful stem cells? Are they going to differentiate into an old state because they're exposed to old blood?
Okay, good question. Andreas?
Yeah, so you all heard the question. Yes, so indeed, if you, in fact, just add serum from young donors, the differentiation and maturity state of our cell progresses to a more mature state. So hepatocytes, for example, the iPS-derived hepatocytes are notorious for failing to express some of the P450s, these enzymes that do drug catabolism when you derive them from iPS. Exposing them to the young human serum actually induces those. So you get more mature cells with the human serum. But we didn't even think about that. That may be another paper to write up that was just a side effect of this. So the maturity in terms of being functional and mature also benefits from the presence of the human serum, but only if it's from young. With the old, you progress again to a dysfunctional state. Did I answer your question? Maybe not? Yes. Yes. Yes, that is a great question. What about the plasma dilution? Is the young serum making our stuff better? So the old serum, right? Mike is on board, right? Old makes you old serum. Does young serum work better than serum dilution? Serum dilution works about as good as young serum. So absolutely everything that Mike said was right. Yes. Mm-hmm.
Great, thank you, Andreas. Give a round of applause for Andreas, please. Thank you. Great job, thank you. Lunchtime. All right, let's go. And it's really healthy. It's all vegetarian. And actually, I had some. I cheated. It's really good.